Hello and welcome everyone. Thank you for taking the time out of your busy schedules to attend this webinar today. I'm Keshav Vasantavara, your host and presenter, and I will be discussing the use of high throughput antibody screening approaches to accelerate the drug discovery process within the therapeutic antibody context. So just so you know, this webinar is a recap of our 9 a.m. webinar that happened today. It was delivered in part by Brady Wu, who is our director of the protein production department. Unfortunately, we ran into some technical issues earlier today, because of which I had to come on board midway through the presentation to deliver the webinar. So hopefully we won't run into any technical issues this time. Excuse me. So quickly about me, I'm the product manager of for protein services and antibody services at GenScript. Prior to joining GenScript, I worked for 7 years in Big Pharma in their discovery group. I was involved in the cloning, expression, and purification of a variety of disease targets to support the therapeutic areas on site. So from my own experience and the experience of my colleagues and our clients at GenScript, I've come to know that recombinant expression is a bottleneck in many labs, particularly recombinant expression in high throughput formats. And so in line with that experience, today's webinar will discuss some aspects around the high throughput recombinant expression of antibodies and the technologies and capabilities that GenScript offers that would allow you to unclog the bottleneck, if you will, of high throughput recombinant antibody expression. And in doing so, I'll also highlight some services that will potentially match your needs and your downstream applications. Essentially, we'll look at a GenScript service that allows the production and purification of recombinant antibodies in an end-user friendly format. And this service combines GenScript's expertise in high throughput gene synthesis and transient expression technologies. And it's designed to help researchers essentially scale quickly from promising leads in order to accelerate their therapeutic antibody projects. Excuse me. So a few housekeeping items before we begin. I assume that you all can see my screen right now. As we move along, if there are any questions, please type them in the chat box. I will answer them at the end of the session. And for those questions that I'm unable to answer for lack of time or ability, a representative from GenScript will follow up to discuss personally. And by the way, this webinar is being recorded and it will be available on our website for viewing later. Okay, so with that, let's move on. So here's the outline of the presentation. Essentially, there are 6 parts to it. I'll do a quick introduction about the drug discovery process, and will then introduce GenScript's antibody drug discovery platform to you. The third part is the consideration, the aspects that we need to consider, in order to identify good antibodies sifted from the bad ones. In part 4, we will focus on the high throughput antibody production, which is a vital part of the antibody lead optimization process. And then in parts 5 and 6, we'll look at some case studies and also capabilities, followed by the conclusion. Okay, so with that, the introduction. So essentially, this slide, captures the, the sense of what was done so far, in this last decade in terms of large molecules, most of them antibodies. So if you look at the figure on the top left-hand side, these are approved antibodies and or fragments in clinical development, many of them approved. If you look at the red ones, they're murine, and the black ones are chimeric, the blue ones humanized, and then the orange ones are fully human, and the green ones are antibody fragments, going all the way from 1986 until 2010. And 22 MAP monoclonal antibodies were approved for therapeutic use in the US, and over 200 antibody candidates were in clinical pipeline as of 2009. Now, I know this data is a bit dated, but essentially it still encapsulates the development that was made in this field. And if you look at the image on the right-hand side, it shows the number of approved antibodies or fragments that are, the different phases of clinical development for 54 antibody fragment therapeutics. The red ones are the ones that are currently studied, and the bluish gray bars are the discontinued molecules, and the yellowish creamish bars are the approved molecules. So approved products accounted for about 20 billion of healthcare spending more than a decade ago, and it's only been growing since. And the market has been growing at 15% per year now, even more. So there is a lot of emphasis on recombinant protein, recombinant antibody molecules, in general biologics as drug candidates. And within that framework, large molecules are trending, and these figures sort of show that effect. So if you look at the chart on the left-hand side, if you look at the market, size, 100%, if you break it down, 13 years ago, biologics constituted only 12% of the market, and the rest were small molecules. And if you look at it every 4 years, up until 2016, the small molecules market has been on the decline, whereas biologics have been growing. And this effect is, partly, or maybe more than partly, actually due to the impending patent cliff. Actually, if you look at this chart on the right-hand side, this shows some of the major blockbusters in recent times, and the ones that have the sales that they generated in 2011. The company is listed on the x-axis, and if you on the y-axis, I'm sorry, and the timeline on the x-axis. If you look at the major approved products, many of them have either gone off patent or will go off patent very soon. So the growth in the biologics market is also due to this patent cliff and patent expiration in many important blockbusters. So with that outlook, in any case, we know that the general R&D, the drug discovery process is risky and it's costly. And to say it very simplistically, the entire R&D process seems like a process of de-risking and optimization. And if you look at this arrow on the left-hand side, essentially it's a very generic scheme for R&D. It takes anywhere between 12 to 16, 17, 18 years for a molecule to hit the market, or goes through discovery about 2 to 10 years, and then preclinical studies, and then clinical, goes into phase 1, 2, 3, and then it's going to be many, many years, and a lot of investment, time and money goes into bringing a product to the market, right? And if you look at this funnel on the right-hand side, essentially it's a small molecule funnel, but also applicable to biologics discovery. You start off with thousands of leads. In case of compounds, small molecules, it's essentially many, many thousands, may also go into millions. And then as you go through the screening process, a few hundred maybe go into preclinical testing, and then a few molecules into clinical testing, and one gets the approval. And it's nearly $1 billion process, invested over many, many years. So that's sort of the landscape. But as much as is the risk, so is the reward, and that's why there is a lot of pursuit in bringing biologics to market. Okay, so with that, within that context, let's look at what GenScript has to offer to support its clients' antibody drug discovery needs. So this is a snapshot of our platform. So we have therapeutic hybridoma services, antibody engineering, in vitro and vivo pharmacology, and antibody production. So essentially, to generate leads, most as you know, therapeutic hybridoma, hybridoma technology is a gold standard. Bulk of the biologics or antibody drug discovery happens. The leads are generated using hybridoma approach, followed by display techniques and or then transgenic animal. So we prepare antigen, do the immunization, transfection, cell fusion, hybridoma screening, hybridoma stabilization for you. And then since it's a murine molecule, then it has to go through the humanization process, right? And which is also something that we offer our clients. We do the humanization, affinity maturation, and then developability assessment. And we also offer in vitro pharmacology services and in vivo pharmacology services, oncology models. And then obviously we have a very robust antibody production platform. We provide transient expression services along with stable cell line development and process development services. So a bit more detail into our platform. I know this is a very busy slide, so I'll sort of deconstruct this for you. If you look at the upper left-hand side, right, and the top half describes the flow of the molecule, and the lower half describes the service that we provide. Okay, so in case of the therapeutic hybridoma approach, we identify hits, and then we go from hits to leads, and then you humanize the antibodies, and then go through the development process, and we get the antibody drug, right? So in case of hits, the idea is to get a desired antibody with your paratope that can identify the target epitope on the antigen, right? And we offer the lead generation service, which is an upstream service. And then as you go from hit to lead, there are various parameters that come into play. The immunogenicity of the molecule, the activity and affinity measurements, developability assessment, and then as you go from the lead antibody to humanization, you go through the antibody engineering process, and then go from humanized antibody into development, and you again need to go through the developability assessment, PK/PD studies. So during this process, in the middle, we look at immunogenicity, affinity, and activity, and then the manufacturability parameters. So we have a bunch of services, antibody humanization, affinity maturation, and the assessment, developability assessment, that provide additional hints to understand your antibody. And then you're going to developing your antibody, and then go into clinical trial and mass production, and we offer downstream process development services. Yeah. So that's essentially it. Okay. And then in terms of the recombinant antibody services that we provide, this is a table that shows the various stages in the the discovery process and beyond, and then the kind of production parameters that you need to use, whether it's transient expression in a shake flask or a wave bag or cell pool or bioreactor or cell line, and then the kind of volumes that you would typically use, and the antibody quantity that you would typically get from these volumes, and then the timeline for each of these stages, and then the guiding price per unit and the QC standards that you would expect at each of these stages. So for example, for a screening phase, we typically use a high throughput transient expression service. Expression culture volume is between 4 and 40 ml. You routinely get between 40 micrograms to 1 milligram of antibody. It's between 2 to 3 weeks from the point of transfection, and then the guiding price per unit is around $300 per variant, and the minimum sample number is 8. And I listed the QC parameters. So it goes for the rest of the stages. Excuse me. Now, since the talk is about high throughput antibody, we'll focus on how to measure the antibody developability using a high throughput approach and the considerations therein. Okay, so when we are considering a good antibody, right, the first part, there are two different focus areas. People look at. One is the functional focus, where you look at the KD for your target antigen, you look at the in vitro or in vivo potency, and then the target specificity and species cross reactivity. And then you have the developability focus, which is the likelihood for the successful development of a lead candidate into a stable, manufacturable, safe, and efficacious drug. So properties such as chemical stability, physical stability, and solution properties become very important at this stage. Sorry. Okay, so for the first part of the antibody drug discovery, hybridoma and humanization, we are focusing on the functional part of the antibody drug. Okay, and this is a real example that we did for hybridoma development for a checkpoint target. So you can see that good leads are really difficult to find. If you start from the bottom of the pyramid, you have to start with somewhere about 1,000 clones. So we immunize 10 mice, titer by lysa and fax, and then 2 animals with top titers were chosen, and 65 96 well plates were generated for hybridomas, and 843 ELISA positive wells of parent clones were identified, out of which 86 clones were confirmed with ligand blocking by lysa, and 26 parental clones were confirmed with binding by fax. Then 26 parental clones subclones, 53 one-round subclones, 28 one-round subclones selected for antibody microproduction, 28 antibodies for single-dose MLRS, and then 12 clones stabilized, which 12 antibodies were selected for ELISA, EC50, IC50 fax binding, fax blocking characterization, and then 5 antibodies were selected for multi-dose in vitro functional assay and epitope binning. So good leads are really hard to find. And then in the next part, we have to deal with the immunogenicity of the antibodies in case they're murine-derived. And the basic method that we use at GenScript is doing CDR grafting, such as putting CDR sequence onto human germline. Of course, we need to do some optimization on that, such as back mutation. And then we can come out with a small library, such as hundreds of variants. And then we can use a high throughput approach to get the best humanized antibody with a good affinity and binding. Okay, so this is a process for humanization. The various steps therein, it takes anywhere between 4 to 5 months. And then when we finish that, we just hit the developability part. And there are several quick factors we can consider, of which I'll talk to you about on the next page. Now, there are 6 parameters listed here, things that we need to consider in developing a recombinant antibody. So one thing is titer. There is a good correlation between the results of transient expression to the downstream stable cell line development. And so starting with a high titer molecule could lead to better success rate downstream. And then of course we also need to consider the affinity and efficacy again. And there are other factors such as stability and aggregation profile, solution properties, and post-translational modification. Essentially, you don't want to end up with a good functional antibody which tends to aggregate very easily, or tends to go bad very easily because it has lots of post-translational modification during process development. And we also need to look at the cross-reactivity to the same family members of the target to avoid any possible side effects, or to the same targets of other species to help you decide what kind of animal models you could evaluate. During a high throughput approach, you can easily measure the first 3 parameters, so the titer, stability and aggregation profile, and the affinity efficacy by binding or inhibition, competitive binding experiments. And that's what most of our clients' applications are when they approach us for their high throughput antibody production needs. So with that, looking at our high throughput antibody production workflow. So at GenScript, we have 2 different approaches you could take as a customer. Essentially, you can either give us the antibody sequence, or provide the sequence and your plasmid DNA. So if you give it a sequence, we go through the sequence evaluation process. We do the codon optimization and gene synthesis, and then cloning, expression, and purification. And we provide the final purified antibody to you. So if you just hand us the DNA, then we still go through the same process. We don't have to do the codon optimization and gene synthesis, but the rest of the steps are the same. And final deliverables include purified antibody and QC data. And QC data includes A280, STS page, and Western blotting. Excuse me. And most of the time, transient expression begins with sequence. So customer provides a sequence. We do the codon optimization, gene synthesis, and then do the plasmid prep. And obviously, you need high quality plasmid. And this slide captures the different kinds of plasmid parameters depending upon the application. So you have research grade plasmid, transfection grade plasmid, and high throughput plasmid prep for your high throughput needs. So if you look at the info in the red rectangle, for high throughput plasmid prep, animal-free cultivation media is included. Advanced animal-free prep is optional. It's more than 90% is superfile DNA. Its endotoxin level is less than or equal to 0.1 EU per microgram. Bioburden analysis is optional. Sterile filtering is optional. And you could use either, for our packages, we use either 10 microgram of plasmid DNA or 30 micrograms of plasmid DNA. Essentially, for the lower amounts of, you need to produce lower amounts of antibody, the 10 microgram package plasmid DNA should suffice. If you need more antibody, close to the milligram range, then you need 30 micrograms of plasmid DNA at least. And this agarose gel on the right hand side shows you essentially the supercoiled DNA in lane 1 and linear DNA in lane 2 and the marker in lane M. Excuse me. And GenScript has invested in a 293 transient expression high density system for high throughput small volumes. So essentially, this is a much more robust system than your standard transient expression systems out there. And it's a mammalian system, uses HEC293 cells, and we have a case study here where we produce antibody I. The antibody criteria listed here, heavy chain and light chain parameters are listed here. The cloning strategy was heavy chain and light chain co-expression, and you can see the STS page on the left-hand side. If you look at lanes 1, 2, and 3, they're reducing lanes using system A, which is the non-high density system. And you see the two bands, about 50 kilodaltons and 25 kilodaltons. And if you look at lanes 4, 5, and 6, they are the reducing lane using the high density system. So you can see it's really night and day difference here. If you look at lanes 7, 8, 9, again, these are the non-reducing lanes of the non-high density system, and lanes 10, 11, and 12 are the non-reducing lanes of the high density system. Again, stark contrast between the regular transient expression and high density transient expression. And the image on the right is a Western blot of the same STS page gel. So you get really robust expression using our high density system, and it's very important when you go, in a high throughput format. You're dealing with small volumes. It's very important to bump up your titers in order to get the material that you desire. Excuse me. Okay. And when it comes to purification, we have also invested in high throughput liquid handling systems. This is the one pictured here. It's a TCAN Freedom U machine. It's very effective machine and has compatible consumables and columns. So this instrument essentially ensures any unwanted human error, and so you get really good reproducibility between batch to batch during purification. As many of you will know, it's very important when dealing in high throughput volumes and production formats. Excuse me. So now with all that background, I just wanted to show you the pricing and some more details of a high throughput G2 antibody service. So the starting material, as I said, most often our customers begin with sequence. They give us the antibody sequence, and we do the cloning expression purification, gene synthesis cloning expression purification. So the minimum requirement is 8 antibodies. Obviously, if you have less than 8, we will accept your request, but we may not be able to order using this catalog price. So we do the transient mammalian system using the high density platform. So if you need it, we have 2 different package options. If you need 50 micrograms of antibody, we can turn it around in 6 weeks, and the price you pay is $4.99 per antibody. If you need 1 milligram of antibody, again, it takes 6 weeks, and the list price would be $7.99 per antibody. And the deliverables include 1-step affinity purified antibody and the QC data. Okay, and a few notes below. So the pricing that we show here includes gene synthesis, subcloning, plasmid prep, antibody expression, and purification. So one important thing to note is the gene synthesis is included in the price. It's the gene synthesis of the variable region. Okay, and this pricing is applicable to human IgG1, human IgG4, mouse IgG1, mouse IgG2A, and obviously not limited to these only. We can certainly build customized vectors upon request. And one other unique feature of the service is that there is no setup fee. Many transient expression providers out there charge a setup fee, but we have confidence in our system, in the success rate of our system, and given our accumulated experience and expertise, we believe that we will be able to deliver a project with a high success rate. So we do not charge any setup fee. And standard QC for this package is, as I had mentioned earlier, was A280, SDS page, and Western blotting. Excuse me. So now with that, we'll take a look at a few case studies and capabilities. And so these are essentially SDS page gels of antibodies that are produced in a high throughput format. Again, you see very nice and clean, reducing and non-reducing lane. So essentially, you would order a high throughput service from us. The report that you get would have images like these, along with other parameters. And for functional assessment, we can do a lot of tests in a high throughput setting, such as binding on BF code or octet, or target specific and cross reactivity, ligand blocking assay by ELISA. And sometimes you have enough sample to run cell-based assays, but more often than not, you require slightly larger amounts of antibodies, more in the 5 to 10 milligram range, 5 to 10 milligram range. But for most applications, typically binding applications and high throughput biophysical characterization, our clients have used this service quite effectively. And we can also do some developability assessment tests. We could do the size exclusion chromatography to detect aggregation. We can do the capillary electrophoresis, STS for charge variants, and LCMS for sequence integrity. And with these assessments and other assessments and functionality, such as in vitro and in vivo assessment, we can give you a pretty good lead. And then you can carry that on to stable cell line development and process development. And speaking of process development, I just have a slide here introducing our process development services. We have very strong experience in stable cell line development in upstream and downstream process development. So for upstream process development, we can do the cell media optimization and screening. We do the fed batch optimization, scale up in bioreactor, post-translation modification optimization. And for downstream process development, we can do purification, process optimization, product quality control, analytical method development, and verification. Excuse me. So with these steps and the associated documentation, we can push your lead to IND much faster. And with that, we arrive at the conclusion. I just wanted to summarize, by saying that, we have the accumulated experience and expertise at GenScript, having purified thousands of antibodies. We've done, literally thousands of IgG purifications. We have experience in delivering many successful IgA production projects. And IgM, as many of you on this call would know, IgMs can be really challenging to produce and purify, but our production team has the experience to do that. We have delivered many successful IgM projects to large biopharma. A lot of bispecific IgG production experience, single-chain variable fragments, bispecific single-chain variable fragments, and Fc fusion proteins. So again, a lot of accumulated expertise and experience to deliver your projects. And then, if you were to ask me to summarize, again, why choose GenScript for your research needs, really 5 things come to mind: quality, speed, price, expertise, and support. So quality, not just of the deliverables, but also quality of the communication and quality of project management. Speed, as you saw, going from sequence to purified material in a high throughput manner in 6 weeks is really fast. So that was just one example. We have a plethora of services that are really the fastest in the industry. So we can deliver your projects very quickly and very efficiently. Price, as you saw for the high throughput project, starting from 1 to 500, going from sequence to purified antibody is very, very competitive pricing, as is the case with many of our other services. And the expertise, as I was telling you, we have a wealth of expertise in our production team. Having produced literally thousands, we have delivered more than 6,000 batches of protein successfully to our clients around the world, more than 2,000 batches of recombinant antibodies to clients around the world, with a very high success rate. And then support, support of our technical team, our production team, the project management team, and the sales support that you get. So your project will be in really good hands at GenScript. Okay, so with that, we are nearing the end of our webinar, and in a moment I'll look at the question panel. But I just wanted to bring your attention to a few upcoming webinars in our series. We have a bunch of webinars. I'm just showing 2 here. The next one is integrated antibody drug discovery, lead generation, characterization, and optimization, which is the next Wednesday at 9 a.m. or 2 p.m. Eastern Time, delivered by Liu Song Yin, who is the Vice Director of Antibody Department at GenScript. And then a week after, we have the webinar Guide to CRISPR-Cas9 Delivery, How to Maximize Your Editing Efficiency, delivered by Alison Mail, who is a teaching fellow at the Memorial Sloan Kettering Cancer Center. So in order to sign up for these webinars, you could visit the link here that I show here. Essentially, it's our webinars page, and you could sign up for these webinars and more. Okay. Right. And any questions? So if you just give me a minute, I'm going to take a look at the questions panel. Okay, so I'll go through a few sort of FAQs, if you will, about this the service that we provide here. I haven't received any questions yet, but until I receive any questions, I'm just going to tell you a little bit about the applications. Some of the FAQs are what kind of applications serve the high throughput gene to antibody customers. Typical applications are for binding assays and for high throughput biophysical characterization. And another routinely asked question is, kind of QC data that accompanies the high throughput service. It's generally A280 SPS page investment blotting. And then if SCC HPLC can be included in the QC, typically we don't do that as default. There is some additional cost to SCC HPLC, and that's determined based upon the request. And the minimum number of samples for this particular service is 8. And another routinely asked question is what is included in the price. It essentially includes the cost of gene synthesis, cloning, plasmid prep, transfection expression, and the purification, and we deliver purified material. Okay, I guess with that, we have come to the end of the session. I just wanted to remind you once again that the slides and recording of this webinar will be archived on our website. I encourage you to visit our website in a few days and take advantage of our webinar resources, not just this current webinar, but also all our previous webinars. And as I said at the beginning, it'll be great if you could take a moment to complete a quick poll right after the session ends to tell us how you like this presentation. Your candid feedback will also help us improve our services. And so with that, we end today's session. Again, thank you all for your time and attention, and on behalf of the entire GenScript team here, we wish you all success in your research. Thank you, and have a nice day ahead.